Welcome to the fascinating world of sponges, the simplest and most ancient animals on Earth. Imagine walking along a beach and finding a dried, squishy, porous object washed up on the shore. For centuries, people actually thought these strange, stationary objects were plants because they do not move around like typical animals. However, these fascinating creatures are the ultimate living, breathing water filters of the ocean. By understanding how they work, we get a glimpse into the very dawn of animal evolution and how life first began to organize itself into multicellular bodies. Let us dive in. Now that we know sponges are animals, let us look at where they fit in biology. They belong to the phylum Porifera. In Latin, this name literally translates to pore-bearing, which perfectly describes their appearance. Why does this matter? Because sponges represent the most basic level of multicellular animal life. They cannot move to hunt or escape, so they rely entirely on their porous bodies to interact with the world. They act as living water filters, constantly pulling in ocean water to extract tiny food particles. This stationary lifestyle forced them to evolve unique internal structures. Building on their simple nature, let us examine how their bodies are actually built. Sponges exhibit what biologists call a cellular level of organization. This is a crucial concept. While their bodies are made of many cells, these cells do not group together to form true tissues or organs, like muscles or a heart. Instead, they are more like a cooperative colony of individual cells living together. Because they lack a stomach or gills, every single cell must absorb its own nutrients and oxygen directly from the water. This is why their internal water flow is absolutely vital for their survival. Since they lack organs, how do they process water? They use a highly efficient water canal system. The surface of a sponge is riddled with thousands of tiny holes called ostia. Water is drawn into the sponge through these ostia. Once inside, the water enters a large, empty central cavity known as the spongiocele. Here, the sponge extracts the food and oxygen it needs. Finally, the filtered water is pushed out through a large opening at the very top of the sponge, which is called the osculum. This continuous one-way flow is the lifeline of the sponge. We just saw the pathway of the water, but what actually pushes the water through these holes? The answer lies in specialized cells called choanocytes, or collar cells. These cells line the inside of the spongiocele. Each choanocyte has a tiny whip-like tail called a flagellum. By constantly beating these tails back and forth, millions of choanocytes work together to create a powerful water current. This current acts like a biological pump, pulling water in through the ostia and pushing it out through the osculum. Without these microscopic engines, the sponge would starve and suffocate. Having discussed the anatomy, let us now look at this cross-sectional diagram to visualize the entire process. Notice the blue arrows indicating the direction of water flow. On the sides, you can see the numerous tiny ostia where water enters the sponge. The water then moves into the large central space, the spongiocele. Look closely at the inner walls. They are lined with the choanocytes, the collar cells we just talked about. Their tiny tails create the upward current. Finally, follow the large blue arrow at the top, showing the water exiting through the single large osculum. Before we look at the physics of their water flow, we must understand why this filtration matters to the world. Sponges are the ultimate water purifiers of the marine ecosystem. A single sponge can filter thousands of liters of water every day, keeping coral reefs clean and clear. Without them, the ocean would become murky with excess bacteria and organic debris. Furthermore, because they cannot run away from predators, sponges have evolved powerful chemical defenses. Today, scientists are harvesting these unique chemicals to develop life-saving pharmaceutical drugs, including new antibiotics and advanced cancer treatments. Now that we appreciate their ecological role, let us examine the physics behind their incredible filtering efficiency. Because sponges cannot move to hunt, they rely entirely on manipulating water currents. They have evolved a brilliant physical adaptation. They ensure that water slows down while it is inside their bodies. This slow speed gives their cells enough time to capture tiny food particles. However, when the water exits, it must speed up dramatically. This high exit speed is crucial because it shoots the wastewater far away, preventing the sponge from accidentally refiltering the same dirty water. To understand exactly how the sponge controls this water speed, we use a concept from fluid dynamics called the principle of continuity. This principle states that the volume of water entering the sponge must exactly equal the volume of water leaving it. The formula is A1 times V1 equals A2 times V2. Here, A1 is the total combined area of all the tiny incoming pores, and V1 is the slow velocity of water entering them. On the other side, A2 is the area of the single large exit hole, the osculum, and V2 is the velocity of the exiting water. If the exit area decreases, the exit velocity must increase to push the same amount of water out. Let us apply this formula to a real-world example. Imagine a marine sponge where the total area of its ostia is 0.05 square meters, and water enters at 0.002 meters per second. The single exit osculum has an area of 0.001 square meters. We need to find the exit velocity, V2. First, we rearrange our continuity equation to isolate V2. Next, we plug in our given numbers. Multiplying the numerator gives us 0.0001, which is the total input flow rate. Finally, dividing this by the osculum area gives us an exit velocity of 0.1 meters per second. Let us pause and think about what that mathematical result actually means for the animal. The water entered the sponge at a sluggish 0.002 meters per second, but it shoots out of the top at 0.1 meters per second. This means the water exits exactly 50 times faster than it entered. This dramatic increase in speed is a brilliant evolutionary adaptation. Because the sponge is glued to the seafloor, it cannot walk away from its own waste. By shooting the water out at high speed, it guarantees that the depleted, dirty water is carried far away by ocean currents. Now that we understand the speed of the water, let us look at the sheer volume of water a sponge can process. To calculate the total volume filtered over a specific period, we use the standard volume formula, V equals Q times T. In this equation, V represents the total volume of water in cubic meters that we want to find. The symbol Q represents the constant flow rate, which is the volume of water passing through the sponge every single second. Finally, T represents the total time in seconds. By multiplying the per second flow rate by the total time, we can measure the broader ecological impact of a single sponge. Let us use this volume formula to calculate how much water our specific sponge filters in a complete 24-hour day. We know its flow rate, Q, is 0.0001 cubic meters per second. Because our flow rate is in seconds, we must first convert 24 hours into seconds by multiplying by 60 minutes and then 60 seconds. This gives us 86,400 seconds. Now, we plug these values into our volume formula, V equals Q times T. Multiplying the steady flow rate by the total seconds cancels out the time units, leaving us with a final answer of 8.64 cubic meters of water filtered in a single day. Let us put that final abstract number of 8.64 cubic meters into everyday perspective. Remember that one single cubic meter is exactly 1,000 liters of water. Therefore, if we multiply our result by 1,000, we find that this tiny, unassuming sponge filters a staggering 8,640 liters of water every single day. Imagine thousands of large water bottles being processed by one small creature daily. This massive volume highlights exactly why sponges are considered the crucial, heavy-duty water purifiers of the ocean ecosystem, keeping the seas clean for all other life. We have covered a lot of ground today, so let us review the most important ideas. First, the phylum Porifera consists of simple, pore-bearing animals that operate at a cellular level, completely lacking true tissues or organs. Second, their survival depends entirely on a water canal system, driven by the whip-like tails of specialized choanocyte cells. Third, by utilizing the physical principles of fluid dynamics, they draw water in slowly to capture food and expel it rapidly to remove waste. Finally, these stationary creatures are vital to the ocean, filtering thousands of liters of water daily. You now understand the fundamental characters of Porifera.